Let's look into what main kind of data analysis we do with this NGS data, where we can perform these tasks. For example, we can do, first of all, we need to do a sequence quality control. We can do a genome sequence assembly. We can take these short reads and we can assemble a genome from that. And when once the genome is assembled, what we can do next is we can identify different coding regions. That's where we do annotations. We can also detect and quantify the gene expression. So if we get the total messenger RNA coming from the cells, we can have an idea about how much proportion of the genes is expressed and what is the amount of that expression. We can also identify variations within different genes and genomes. We can identify different variants. So in this figure, we start from the beginning and we just give an overview of this data analysis. So for instance, we come up with a sample and then we do some genomic extractions. We prepare cDNA libraries and now everything becomes a DNA. And after that, we do that shotgun sequencing. So we break those segments into those smaller fragments. We attach adapters. And now the libraries are ready to be put into different sequencing platforms, where we can have a second generation sequencing or next generation sequencing, where we amplify them and then we sequence them. Or it can be directly third generation sequencing, where we can take these sequences and we simply sequence them. So within the sequencers, maybe those nucleotides are put together and then we are getting some images, and in the end we come up with some reads, which we call them as raw reads. So which reads are actually in FASTQ format. So what we can do with these raw reads, we can take these reads and we can do some read cleanliness or quality control. Now we have seen those fragments where we have those green and red colors. Actually they were adapters. So we actually remove them and now we have clean reads still in FASTQ format. So quality assurance is done. We can put them together and then we generate a reference. So once we put them together, the assembly, this assembled genome can be used as a reference for future studies. We can take that reference, or maybe we can take some previously existing reference and we can map these reads on that reference. And if we have a reference genome, we can do its annotations, and GFFs are these different annotation formats. When we align them, we come up with this thing which we call it as BAM. These are binary files. Here we have the raw BAM files and we then process them, and what we can do with them is we can call SNPs from them, and we can also do from these BAM files, we can also study the differential gene expression. We will look into the conversion of this BAM files into another format called as SAM files in a moment. So what interesting tasks we can do further? First of all, we can look into the sequence length distribution. So if our sequencer is producing the reads, for example, 100 base pair in length, and with this length distribution study, we can see that whether we are getting the appropriate length fragments or there are some issues. FastQC and PRINCIC, these are two important tools which we will include in this discussion, and they are used for quality assurance. Let's look into the sequence length distribution. We are looking the results coming from PRINCIC, where we see that these lengths, they range from 100 to around 539 base pairs, where we can see the mod is, the sequence length is around 350, somewhere around this. We have many reads which are of this much of the length as far as this segment is concerned. So this is how we can have a rough idea whether our sequences are corresponding to the lengths which are specified by the sequencer. Same way, FastQC gives us this graph in this shape, but it does the similar thing. As far as sequence quality scores are concerned, that's are also important concern, that sequences should be of good quality. Normally in the softwares, Q20 is mostly used, so minimum score should be around Q20 and it should be between score of 20 to 40. Let's see, here we have the output coming from PRINCIC. where we are putting these quality scores. This representation is called as box and whisker plot. We will cover in some statistics primer later on, but just get a general idea. So these boxes, they represent where we have those middle 50% of the values, and then obviously we have a lower 25%. Let me draw it somewhere here. So this representation is called as box plot, where we can plot all the values, and these numbers, if we plot them together, they become a distribution. So in the middle we have middlemost value, or the median, so that splits data into two halves, and then we have the data in quarters. So here is lower 25%, here is then middle ones, and then here is 50 to 75th percentile, and then we have the total upper 25%, which is right here. So if we look into it, we can see that the score ranges from 30 to 40 as far as this left hand side is concerned, whereas the median score is somewhere near to 40, maybe 38 or something. If we start moving from left to right, so down below we have different sequence positions. So what they are doing here is they are taking all those reads, putting them together, recording their scores, and then looking at position number 1, what was the distribution of the scores, and then they keep moving till they reach towards the end of those reads. So all 540 bases and their scores. So if we look into the pattern which we have observed over here, that towards the left hand side the boxes are quite close with one another, medians are higher, and if we move along right hand side, the scores are coming down. That is the common feature in those sequencers, next gen sequencers. They start with good performance, and as long as they continue, the chemicals, they go over and then they get consumed, and obviously the machines, they get heated. And one drawback of this NGS technology is that the sequences which are produced, they are bad at this 3' end. Same trend we see, a little bit clear picture coming from FastQC. Again we see that the sequencing quality is good as far as these starting reads are concerned, and it's bad towards the other side. So what we can do here is, while looking into this data, maybe we can go up to some level. So for example, from here where we have the minimum scores are 20, and below that we can see that there are some reads which are still performing bad. So maybe we can clip those regions, and we may not include in the future analysis. As far as the nucleotide distribution is concerned, it is generally expected that this distribution should be pretty even. So all bases, they should be represented in an equal proportion, more or less 25%, or with a probability of 0.25 each, 1/4, since there are 4 of them. So let's see how these distributions they look like. They are correlated with different regions. So different regions where we have, say for instance, if we have some promoter regions where we have lot of GCs, so at that regions we might have the proportion of GCs higher as compared to rest of the nucleotides. So here we see the nucleotide distribution. Actually it's a proportions kind of a chart, where we see that there are different colors labeling different nucleotides. So A is green, C is blue, then we have T's which are red, and then we have G's which are black. So let's see what is the proportion of different nucleotides. If we take position number 1, we see that the green lines are higher, so it's kind of around 60%. We have A's here, a similar trend. Here we have blues, where we have more proportion of C's. So looking into this pattern and correlating with different regions can be an interesting study, or sometimes we can have some expectations where we might expect some specific patterns of these proportion of these nucleotides to be present. So putting them together in a graph like this can help us decide the quality of the data or get some specific patterns out of it. This can be seen in FastQC, like these line graphs. So looking into sequence length distribution, quality of the bases, relative proportion of different bases in our read data can be an interesting step in quality assurance studies.